Hi, everyone, and welcome to the Space for Women show. For some of you who are here tuning in first time, the Space for Women show project was launched almost a year ago in support of UNOSA's Space for Women network to address Sustainable Development Goal 4 and 5, and specifically to promote space technology. Space Connects, a Space for Women show, was a creation of UNOSA Space for Women initiative and focused on awareness raising about key issues related to gender equality in space and STEM and contributing factors to gender inequality in these fields. Today's episode 37, Space Microbiology, the importance of microbes for space flight, will discuss a new frontier for space biology. And we hope new STEM research will evolve from there as humans move out of low Earth orbit. So in part one, we will discuss space microbiology overview with Dr. Christoph Lahtz, a biochemist and space biologist. In part two, Professor Dr. Jamie Foster from the University of Florida will then foster space for women through her own story, and she will share with us how microbes maintain host health. In part three, then, the second half, we will have a Q&A and an interactive discussion. So you feel free to ask questions to the audience, to the panelists directly. So without further ado, let me introduce you to our moderator today, Professor Dr. John Kiss, who was recently awarded by the Committee on Space Research, COSPAR, for a significant contribution in space research. So the floor is all yours, John, Dr. John.
Well, thank you very much. It's a real pleasure to be here. We have two very distinguished and exciting guests with us. And we're going to start with Dr. Chris Lahtz. He's originally from Germany, and he's in Los Angeles now for a postdoc. He's told me he's always loved biology, chemistry, physics, and space, all things space. He wanted to become an astrophysicist. He has a master's degree in biochemistry and a PhD, a doctorate in cancer epigenetics. And with that brief introduction, let me turn the floor over to Chris.
Yeah, hello, everybody. Thank you for the nice introduction. Today I want to talk a little bit about space biology in a more broader view. And today I want to talk a little bit about myself before. Dr. John Kiss already said, so I always loved in school, basically biology, chemistry, physics, and space. I was also only good in biology, chemistry, and physics. So that led me to the idea, man, I would like to study astrophysicist, and I wanted to do that already with the age of 12, but then I had a few incidences with very boring physicists in a different field. That was a little bit sad. That could have led my path somewhere else, but so I decided then to study biochemistry in Germany. I mean, I live in Germany, so I studied biochemistry there, and then I did my PhD, doctor in Germany, cancer epigenetics, because I was always interested in genetics too. And then after that, I moved to Los Angeles for my postdoc. And right now, as a night job, I combine my love for biochemistry and space. So that's why I'm here today. So I want to start with a short overview about my talk. I want to give a short introduction about microbes and the differences in space environment and what happens there, the influence, then about the health risk for astronauts and the opportunities we have with bacteria and microbes in space and for space colonization. And then I want to sum up with my key points. So actually our planet could be called a planet of microbes because 60% of the biomass of Earth is actually consists of microbes, even when we don't see them. Microbes are the oldest and the first life forms on Earth. So they are likely 3.77 billion years old. There are some research they are much older, but this is so the conservative assumption. And because of that, microbes evolved in all different corners of our planet, so even very extreme environments they can live in and thrive. So you can see that here too. We have here on the bottom the hypothetical last universal common ancestor, where all life forms, what we have right now, all branches is split off. And here you can clearly see why we could call Earth as a planet of bacteria, because on the left side in blue and in red we see bacteria and archaebacteria, which are all microbes. And far on the right side we see animals, fungi, and plants, what we see in our daily life. So, but these are only very little branches of life. What is out there? So it's a lot of microbes we have on Earth. So the differences in cell architecture are also very large. So we have here on the bottom one.
Oh, it's here.
We have here on the bottom one a typical bacteria cell, like bacteria and archaea, called prokaryotes, because they don't have a nucleus. As I said, they are the oldest cell type. They are very small, smaller than 0.1 to 5 micrometer in size, and very simple. So as I mentioned, no nucleus, they have no cell organelles, they have a circular DNA here in the middle. The DNA is here in yellow. They are always unicellular, and we have only bacteria and archaebacteria here. So in contrast to that, the eukaryotes up here, that's an animal cell or a plant cell, they are much different. So they evolved from prokaryotes 2.6 billion years ago. They are much larger, with 10 to 100 micrometer in diameter, and much more complex. And the first really multicellular life form, like plants or animal, that was actually 600 million years ago. Compared to that, Homo sapiens arrived on Earth 200,000 years ago. So it's a very long time. So they have a nucleus here in the middle, and they have cell organelles like the Golgi apparatus, like mitochondria, and the endoplasmatic reticulum. The DNA is linear and organized in chromosomes, and they can be unicellular or multicellular. The difference to plant cells largely is chloroplast, and they have a cell wall instead of a cell membrane. So since life evolved on Earth, life never experienced, of course, space conditions, which is microgravity, so the absence of gravity, and much higher radiation. So what happens actually when we bring microbes and astronauts to space? So we talk about microbes today. So what microbes do, they really thrive in space conditions. So they have a reduced lag phase, what means that the break between cell divisions is much shorter, so they can grow much faster, and they can also grow much denser in density. So, and because of the radiation, we have a lot of DNA damage and mutation rate, what I can show here. So we have two ways how the radiation can affect the cell. We have here the direct influence, what affects the DNA. There's a damage if that cannot be repaired. We have a change in the genetic mutation that can lead either way to cell death or mutation, what could cause a loss of function or a gain of function. And also the indirect way, where the radiation creates radicals inside of the cell. That happens most of the time with water and other molecules, and these actually can damage the DNA again and cause cell death and mutation. So what happens too is that the microbes get more drug resistant in space, and the virulence is also increasing. The reasons for that, as my knowledge, is kind of subject of research. And combined with the immunosuppression with astronauts, it actually is it's an increased health risk for astronauts in space and in space colonists. So you might think you don't want, because of that, you don't want microbes in space, but actually microbes are very, very useful and helpful. So we could use this drug-resistant and virulence as a possibility to get to develop better antibiotics and vaccines. So, for example, fungi and also bacteria, they produce in space different secondary products because of microgravity, and it could happen that we get a new antibiotic there, and also for the vaccines, it's a good research. And also bacteria, fungi, and cyanobacteria especially are very useful for vitamin production. Vitamin production in our intestines, of course, the bacteria, we need that. Water recycling, air decontamination, and waste management. So here's a very, very simple idea of a space colony. So we have the astronaut or the colonist here in the middle, and he needs oxygen and food, and he produces organic waste and CO2. So the plants, what they bring with them for a space colonization, could do the food and the oxygen, but it's also a very good idea to have spirulina, what is a cyanobacteria, what produces also oxygen and actually can be used as food. And spirulina can also use for cyanobacteria, can be used for biofuel. It's a good idea to have another energy source, since on Mars, for example, we have only 40% of sunlight. So, and also we produce organic waste in space, and we have not the luxury to waste anything in space. And at the space station, it's okay, but further away, it's getting more and more critical. So we need to recycle everything what we have there, like the water, ammonia, and phosphate, and everything. And need to channel that back to breed plants and bacteria, what we can have, what we can grow there. So one example is here. This is actually a research done from the DLR in Germany. They tried to clean urine with bacteria. So what they did is they had here a column with very porous filter material, and they made bacteria settle in there, and they let flow on a very slow rate urine over this, and the bacteria kind of decayed the urine back to nitrate and as a plant food. So that worked. It took kind of a little bit longer than they wanted to be. It needs to be optimized to be very efficient for cleaning up on a space station, or a space station wouldn't really work. We need to, because that works with gravity. But as a principle, it works. It needs to be optimized. And also cyanobacteria, as I mentioned, are very, very important on a space station, on a space colony for oxygen recycling or food and biofuel production. And here's another Mars colony, how that could look like. So we have here a pool with its cyanobacteria, and we get the CO2 and the nitrogen and the nutrients from the regulus inside, and we can produce food and oxygen, biofuel, and nutrients for greenhouses. So my key points are now, I want to sum that up. So bacteria are the oldest, the simplest life forms on Earth, and they can grow practically everywhere. The cell structure is very different. Under space conditions, bacteria grow faster, and antibiotics don't work as much. They're more virulent, but we can use that to develop better antibiotics and vaccines. And furthermore, they are, besides that health risk for astronauts, they are very useful for space and colonies, for vitamin production, water recycling, air decontamination and recycling, waste management, and food production. Yeah, thank you very much.
Thank you very much, Chris. We'll hold with questions and discussion till after our second speaker, Dr. Jamie Foster. And Jamie is a friend and a colleague of mine. She's a very distinguished scientist and has many accomplishments. She's been involved with NASA and spaceflight research. She's also been very active in the American Society for Gravitational and Space Research in many roles, editor of the journal. She's also president of the organization. Currently, she's on the faculty of the University of Florida in microbiology. So let me turn it over to Jamie.
Thank you so much for coming on your Saturday and spending time with us to talk about microbes in space. It's a topic that I love, and I think it is a very exciting time to be a young person working in the field of space biology. That's kind of what we're talking about today. And I just want to introduce myself, a little bit about myself here is I actually have a PhD and a bachelor's degree in zoology. So how the heck did I go from a zoologist wanting to be a dolphin trainer at SeaWorld to someone who gets to do experiments in space? And I was actually the first person in my family to go to college. So this was not, I didn't have a long legacy of academics in my family, and we were very much a very blue collar family, but I got the chance to go to college, and it was really a mentor that really changed my life. Dr. Lynn Margulis, she was a professor at the University of Massachusetts where I went to school. And she's actually quite famous for being a proponent of the endosymbiotic theory, that cell that Dr. Chris was showing us, that all those chloroplasts and mitochondria, that all those organelles were actually free living organisms at one point in time. And so Lynn was this really big scientist, and yet she took me under her wing, and she helped mentor me when I knew nothing about going to grad school or doing research. And so I can't express how important it is to have strong mentorship and to be a mentor to others, and also to reach out to your faculty, your professors, or people you might be working with to take and ask for help if you want to have a career in this field. But as I mentioned, I went on, and I have a big picture of a squid here because I loved the marine sciences, and I said I wanted to be a dolphin trainer at SeaWorld. But I found this research opportunity through my professor, Dr. Lynn Margulis. And so I started working on symbiosis, how microbes were affecting the health and the development of animals. And then that spread to these rock-like structures here. And those are called stromatolites. And these are the oldest known ecosystems on the planet. So you might be wondering, what does a squid have anything to do with these rock-like structures? And the answer is microbes. Microbes help, they've kind of pushed the evolution of animals. They've pushed the evolution or the change in the environment. And I'm fascinated by how microbes are talking to the ecosystem around them, and in return, how the environment is shaping the evolution of life on Earth. And that information is what we're going to need to move on and to live beyond our own biosphere. And then what I did is, once I got my PhD, I really, really had this drive to be an astronaut, just like Dr. Chris, and work in the field of space research. And so I wrote, as a postdoc working at the National Institutes of Health, I wrote a fellowship and got to work at NASA. And then I left my job, my fellowship, a little bit early because I got this opportunity to be a professor at the University of Florida. So let's talk a little bit about some of the things that I'm interested in, and things that are really driving the field of space microbiology. As I mentioned, it's an exciting time. We're starting to think beyond the International Space Station, and we're looking to the moon first, because the moon it's only a couple 100,000 miles away. We can do there's some exciting, a lot of new scientific research happening on the moon. And so if we're going to spend longer durations in space, we really need to understand not just how human physiology is changing in space, but what's happening to the microbes. So we call that the microbiology of spacecraft or of space research, the microbiome of the built environment. So the built environment is basically any kind of structure where humans spend time. And we have become an indoor species, we really have. And so the microbiome is all the microbes that live in the nook and crannies of all the spacecraft, or it could be your kitchen here at home. So understanding the microbes that surround us is incredibly important. And just some fun facts about what's happening in your own home. Um, there are a lot of microbes in your home, and that's a good thing, actually. As you can see here, I have some fun facts here about the average home. If you just sweep the door sill of your house, you're probably going to find 7,000 different bacteria, a couple thousand different fungi. And so there is a diverse, rich community living in your house. It's same with the hospital. And as we interact with those environments, a lot of genetic material is being exchanged. But it's unclear as how viable or how many of these microbes are living, and it's precisely how are they interacting with our bodies. So it brings up some big questions in space microbiology that people are thinking about, especially now as we go beyond low Earth orbit and we're going to spend maybe years at a time in space, not just a couple weeks or a couple months. And so what microbes are we being exposed to in those kinds of spacecraft settings? What's driving the populations? What's controlling those microbes and how they persist in that environment? And then what's going to happen? Are the microbes degrading the material around them? Are they changing the materials? Do we need to be concerned about that? And so these are some big questions that we have to tackle for long duration spaceflight. And then it comes down to us too, what they may be interacting with the habitats, but also how are they interacting with our own bodies? And so that's another big question that I work on is what's the balance between health and disease? Microbes are essential for our health. And we know certain species or certain taxa can cause, all sorts of problems. And if you have the right microbiome, you can prevent things like asthma, or you can control diabetes. There's so many health physiological effects that are due to perturbations or changes in the microbiome. So the squid project, I'm still working on squid 30 years later here. And I'm very interested in how the microbes, so what's valuable about a squid in this symbiosis is they have a single organ where one bacterium lives. And so we, instead of trying to understand, the thousands of different microbes that might be out in the environment interacting with our cells or our airways or something like that, this symbiosis with one host and one symbiont, we can really start to understand and really manipulate how microbes are communicating and changing the host tissue. And we're looking at that in the space environment. But there's a lot of other questions. So I'm interested again in how microbes are affecting animal health, and we want to translate that to larger scale, to humans. And so right now, there's several ongoing programs on the International Space Station to really address those questions that I brought up earlier. What are the microbes on the spacecraft and how are they changing over time? And so there's a new program called the Microbial Tracking program that's happening. There's been several iterations on this. There's going to be a Microbial Tracking 3 very soon. And they are literally swabbing different parts of the spacecraft, looking at how those communities are changing as astronaut crews change and also as over time. So how stable are these populations? How are they interacting with the materials? And do we need to be worried? Are there pathogens out there? And how could we manipulate that? And so it's pushing the field forward in terms of as we go to the moon, we're not going to have astronauts there all the time. So we're going to have to automate a lot of these processes. So maybe some of you out there are interested in technology development where we can start to begin to automate many of these things we take for granted here on Earth by being able to just go over to a corner and swab some microbes. We will now need instrumentation and computers to help with that. So we need to start really monitoring microbial communities in space on a more real-time level. And the next step then, could we use some of these microbes that we know are beneficial here, on farms or in our backyards? Can we control populations on the spacecraft to make sure, almost like a probiotic for a spacecraft? Can we use, or what I call the probiome here, can we use microbes to control and mitigate any potential problems in our water systems or those bioregenerative life systems that Dr. Chris was talking about? But the bottom line is we need you. We need more people. I mean, we need zoologists apparently, right? I'm a zoologist working in this field. We need computer scientists. We need mathematicians. We need geneticists. We need all of you to participate and get involved in the space program, whether that's through space biology questions about supporting and keeping humans alive, or maybe it's more astrobiological questions, trying to understand how do microbes evolve under different stresses like cosmic radiation or low pressure and things like that. So I challenge all of you to get more involved and hopefully become a space biologist yourself. So with that, I will turn it over and for questions, and thank you all for your attention.
Thank you, Jamie, for a really interesting talk. Let me just quickly follow up on one of the things Jamie said, I think is really important in terms of identifying mentors and career development. In a previous show that I was on, I mentioned that I too am a first generation college student. In fact, I'm an immigrant to the US. I came when I was 4 years old from Hungary. But I think Jamie's point, and I didn't know this, but I didn't know about her mentor, Lynn Margulis. That's really pretty cool and very impressive. It was a transformational figure I think in biology. But it's, I think all of you out there who are looking at careers in space or science or STEM, it's really good to have identified mentors, go up to them and say, hey, I'm really interested in this. And most people respond positively. So I couldn't help but add my own comment on that. So I'm really glad you brought that up, Jamie. So at this point, we'll ask some questions and we'll get some questions from the audience. So the first question is, should we be concerned about introducing terrestrial bacteria and maybe other microorganisms into new environments such as the moon, Mars? And in fact, I think Jamie was at NASA, had this lunar symposium, and this topic came up of planetary protection. So Jamie or Chris, could you comment on that?
Well, I can just reiterate what Lisa Pratt. So Lisa Pratt is our planetary protection officer for NASA, and we were talking about this very week about how do we make sure that we do not contaminate. We want to find life on Mars, but we don't want it to be E. coli, right? So we want to make sure we're taking the necessary precautions to do what's called it's called planetary protection. We want to protect the place where we are going to. That is to prevent forward contamination. And then on the flip side, we want to prevent backwards contamination. We don't want to bring it. We do not know if there is life on Mars. We're pretty sure there's no life on the moon just because of its conditions, but we do not want that exchange happening and potentially skewing anything. So Dr. Pratt led a great discussion about how do we as a community set guidelines to make sure and very stringent criteria depending on where we are going. And the target depends on the potential for habitability. So the moon is very low, it has no atmosphere, it's exposed to enormous amounts of cosmic radiation and solar wind and weather. So it's probably a low target in terms of potential life. But Mars, that's a much higher risk target. So there are a lot of stringent activities to prevent microbes, because they get under tape and they get in the nooks and crannies, and they could be protected from solar radiation. But there's an enormous field, and it is enormous concern for the community about planetary protection. And I'll let Dr. Chris add some more words to that.
Yeah, thank you. Yeah, I mean, I think they really cleaned the spacecrafts with gamma radiation, but there are very extreme bacteria out there what could survive that if they are there. So the cleaning process is very intense. That's why they run around with all these cleaning suits and so, but cannot be 100%, I guess. I think they found also recently, not recently, but they found a strain what survives autoclavation, so very, very high temperatures.
Well, they don't
pressure.
autoclave. Just to let you know, they don't, other than Viking back in the 1970s was the only instrument, the Viking 1 and 2, they were the only ones that were autoclaved or
Yeah.
completely sterilized, I should say, because it's
Yeah.
extremely expensive to get electronics to work
Yeah.
autoclaving. So like you said, they do cleaning, but they do have a threshold. You can have, because they're not sterilizing it, there is a
Yeah.
certain allowable amount of microbes to hitch a ride.
Yeah, and also what I want to mention, what went to the media a couple of months ago, I think there was a Chinese spacecraft was crashed and was releasing tardigrades on the moon. So that is also, that was an accident, and we contaminated the moon. I mean, tardigrades, they are pretty tough, but they will not thrive on the moon. They will be there, maybe they survive, but even that is doubtful over the years. And yeah, but yeah, we need to be careful also when we have experiments to expose the Mars, even when we bring it in good faith for what I mentioned for some applications on the Mars. But yeah, it's very difficult to contain microorganisms in general. Yeah.
And I think even on the moon, Jamie, there was a talk about looking at what the Apollo astronauts brought back from one of the robotic landers, that there was
there was like
thought,
a Streptococcus cell that obviously survived the clean room process and the transport, and
yeah.
so even though that was a short duration mission and that microbe was probably protected, being in I think it was in a camera case or something like
Right.
that, found a viable microbe afterwards, but it was probably a hitchhiker the whole ride. So yeah, it definitely just raises the threshold of and it's probably going to influence how we explore Mars when humans finally get there, because we're probably, we are walking petri dishes everywhere we go, everything we touch, hence why we have a pandemic right now. So we're probably going to be restricted to where we can go if there are pristine sites on Mars or something, and we won't be able to just traipse around everywhere, and as long as there's the question of, is there life, indigenous life on Mars?
Okay. Another question, should we be concerned about inadvertently exposing humans to new forms of microbiota, for instance, on Mars?
Well, we don't, I wish, right now we only have an N of 1. There's only life on Earth. So we do not know if there is life on Mars. But absolutely, there probably would be quarantine. The astronauts that came back from the moon were quarantined for a couple of weeks when they were brought back. But absolutely, and that gets to the other half of that planetary protection in reverse contamination. Are we bringing back anything potentially hazardous that could be threatening to life on Earth? And so that is the other side of the coin for planetary protection, and that is thought about extensively, even though currently at this moment in time there's no evidence for life beyond Earth.
Chris, do you want to add anything?
Yeah. So there are some stuff what you can think about. The beginning of life, we are not sure where the life originated. Maybe that was Mars, maybe that was Earth, and there could be an interchange between both planets in the past with asteroids and stuff what could have survived. So the question is, if there is life on Mars, is that related to us, or is it totally different? And when it's totally different, it could affect us, could not make us sick, but as we know, life has to, or bacteria, pathogens have to be kind of have grown on and have to have the same evolution like we had to make us sick. For example, we don't get the dog flu, at least when it's not mutated as much. Now we got another flu, but it's not necessarily when that stuff from Mars originated on Mars would make us sick, even when it's originated the same. The evolution was a long time separated, so it could make us, but it's not clear. So it's very unknown if there is life. I really hope there is life on Mars, but yeah, I'm sure we need to protect ourselves because we just don't know it. Yeah.
So
I just want to add, can I add something to what?
sure, go ahead, Jim.
Said? So I think that you touched about the origin of life and could Mars have its own indigenous life? And I think that is a profound question. If you were talking about lithopanspermia, this exchange of microbes between worlds, and we have about 50 Martian meteorites here on Earth, so that we have strong evidence that that pieces of the planets are being exchanged between Mars and Earth. I think the more profound question, as you said, is did life on Mars evolve independently? And if life evolved independently twice in one solar system, then I would say that the whole universe is teeming with life. I mean, if it could happen twice, I think that's the more profound question is is there life on Mars, but is it similar to life on Earth or a completely unique independent evolution? I thought just when you said that made me think about that question.
Yeah, I really think that is a fascinating question. So look around, maybe we're all Martians, right? I mean, we could have started from, I don't have anyone to look around here, but
or Venus. We started on Venus and then no longer habitable. I mean, that's a possibility as well.
I know very little about this area other than what I've read, but like Jamie, I think it's a very profound, fascinating question. I do know, and I've checked with some planetary scientists, astrobiologists who will say that the conditions on early Earth versus early Mars, that Mars actually was more favorable for the formation of life. That's another, I guess, theoretical discussion. And that was
there's a lot of good evidence. Yeah, there's a lot of good evidence in terms of the fossil water history and in the mineralogy on the surface of Mars.
Yeah, there's a whole bunch of questions in the chat, but that theory of transpermia, of
lithopanspermia,
yeah, had come up. So another question we have is, could we grow our own antibiotics on Mars like the way penicillin was initially discovered?
I would start with the moon first. I think it's wonderful if we all go to Mars. I hope one day maybe someone in the audience will have an opportunity to take a Virgin Galactic to Mars. But I really think we really need to use the moon as a test case. And there's a lot of good science questions there, but I would work on the moon first before Mars. That would be my suggestion. But absolutely, we know so little about how we were going to have to do pharmacology, and Dr. Chris might be better at speaking to this myself, but we are absolutely going to have to grow our own natural products and be our own pharmacy. We cannot pack a gigantic picnic basket and take it with us to Mars. We are going to have to start to grow our own foods and be a little more resourceful. There's already 3D printing in space. I think that will be absolutely something we have to do on the moon, not just on Mars. And it could really benefit us here on Earth if there are new ways, new stresses, new products that the microbes might be expressing that we wouldn't have known otherwise if they weren't in space.
Chris?
Yeah, that is actually exactly my point in the past too. When I say everyone is talking about, yeah, we go to Mars, we go to the moon, and we build a city, and yeah, a little bit of radiation, we go under the ground and so on, and yeah, but nobody's discussing. So how we live there, we need to clear out the biology there, and that's why yeah, we cannot have all the time trade spaceships coming with antibiotics and stuff. So we need to produce everything there. So and as in the past, I think beer was the first biotechnology of humans, right? So they used, it's actually pretty complicated to make beer when you don't know how to make beer. So yeast is also, it's not a bacteria, it's a fungi, but so we need to relearn that basically, do everything with microbes and fungi ourselves. So we have the technology to genetic engineer new bacteria to put something in, like plasmids, so the blueprints of stuff what we want to make there. But yeah, there will be a lot of bioreactors on Mars and Moon to produce what we need there. So yeah, and nobody's talking about that. So it's we need to raise the awareness for that.
Chris
Just to
raises it. Oh, go
Yeah.
ahead, Jamie.
No, I was just going to add, if for people who are interested in kind of what the commercial applications are for a lot of this pharmacological, there's a lot of really neat experiments being done on the ISS National Lab. So the International Space Station is now a national lab for the United States, and half is international, and then a part of it is for American research. And there's a lot of companies from all over the globe doing science on the International Space Station, looking at ways to perfect drug function. And you might want to look at it used to be called CASIS, but for people who are interested, they could look at all the different companies that have done experiments on the International Space Station, because it's pretty impressive. And you can see all the material science, the material engineering, the pharmacology that's being done right now, right as we speak, 250 miles up.
Yeah, it's a really good point. Chris, I think, raises a really fascinating point as well in his previous comments. If you look at sort of the popular imagination, when we're talking about mainly Mars, but I guess also Moon, the focus is, okay, guys and girls, we got to build this gigantic rocket. And that is absolutely true. But really, the problems to colonize Mars are biological questions, such as the ones we're talking about. And that's kind of a lot, it isn't as exciting as building the big rocket for some reason. But, I think things like, what kind of medicines, what do we need to pack? I'm a plant biologist, so, maybe we could genetically engineer plants for some medicines that we might need, just as well as we could do that with bacteria. So really, I think the questions for space exploration are biology, and that's where it's at. So I'm really glad Chris raised that issue, and I see Jamie agreeing with me, so that's good.
I know completely. We have to have plants, and we have to bring our ecosystem with us as best we can. And again, that's why I'm suggesting we go back to the Moon more readily, because there you can do short-term 6 months experiments rather than having to rely on a 2 or 3 year transit. So there's just a lot of, going to the Mars right now would be a showstopper with just the amount of radiation and that a person would, or plants, or the animals, the food that would have to. So I think that going to the Moon is just a smart way to go and test all these questions that you're bringing up.
So Yara, there's been tons of interesting questions in the chat. Do you want to pick out a question that you found intriguing?
Someone asked, I recently read about how some microbes that have been long buried in the glaciers are now at risk of re-entering the ecosystem because of climate change. Can current methods also prevent those older microbes from being introduced in Mars or Moon when those microbes get unlocked?
So basically, you've got these kind of fossils, living fossils hidden away in some of the ice, and there's ecological concerns about how, when they get in, probably, to be honest with you, if there's so much microbial diversity, they probably wouldn't have much of an impact, because there'll be so much competition for resources as they get released. or if they've been spending a million years in, you know, in the ice, the briny ice crystals of they may not survive being able to exist in a higher temperature environment if they've been all of a sudden released. So there's some really great, you know, microbial ecology studies on that. Or are you talking about Mars? If we release something on Mars, I'm not sure what the person was referring to. But
Question seemed to be on like the Earth and Antarctica and
yeah,
things like that with global warming is the way I
yeah, there's
understood it.
some fascinating work done on the ancient microbes that basically live in the briny crystals between, you know, ice structures. And absolutely, we have to study and understand and sequence those organisms to look at their genomes and see if they could potentially be a threat. Because I would imagine the growth of those organisms is very slow. So I have a feeling we could probably have a head start on them. They've probably been hunkered down for a long time in the ice.
Thank you, Dr. Jamie. There's a question from Dr. Egbert from the Netherlands addressed to Dr. Christoph. Do you think CRISPR gene editing technology can be used to upgrade human radiation resistance in the future?
So that's a very interesting question. There is they figured out recently about this gene, what tardigrades use to protect the DNA, and you could think against radiation. So you could think to use that technology to make astronauts also a little bit more resistant against radiation or other genes from bacteria. It sounds like science fiction. It's probably possible in some way, but not right now. There are also a lot of ethical questions because we would create a new human species right there. But that it goes into all cells, and they can pass it on the next generation. And if we have that, so we have a little colony on Mars, and we make the initial set of humans resistant against radiation, for example. And with this technology, what is actually very difficult, because you should start from scratch, like when you from the first cell. Otherwise, it's very difficult to have a somatic change in our cells with that. But so you could get that in the next generation. Then you would create different human species over time. And I think we are not there yet with the technology to do that in general. And even if we are able to do this, there are ethical questions too. The question is, what is more useful, to survive on Mars being genetically engineered, or keep ourselves? There's an idea, I think that is pretty famous, but I don't remember the name, who said that to colonize space, we can set up a spaceship with humans, which has no gravity in the spaceship, and instead of legs, we have a second pair of arms engineered, and this spaceship drives around space, and when it finds a planet what could be habitable, so they engineer especially humans for that planet. So that is a very science fiction thought about that. But
Thank you, Dr. Christoph. Thank you.
Yeah. Can I add something here? Is I think just a broader thing. You know, we talk about science fiction, and I think good science fiction has been very predictive of some things. So one example I always found intriguing is, you know, in 1900, Jules Verne predicted that there would be a spaceship from Central Florida that would be launched to the moon. Now, he said there would be 4 people on board. Well, he was wrong. It was only 3. But, you know, still, you know, there's different kinds of science fiction. And, you know, that's another fascinating topic. One question that came up that seems interesting, food growth on Earth has significant amount of water, waste, energy, and water. What technologies do you see improving this for space and also improving the Earth situation?
Yeah, I mean, John, you should probably take that with regards to your plants.
Well, I actually think, yeah, of course, that's plants, but I think it's actually more like the Melissa project and like more microbial. So I'm going to give it to you, Jamie, since you are a
Sure.
distinguished speaker.
I've actually been looking at, so those stromatolites, those rock-like structures that I showed you in one of my first pictures, they are actually like microbial biofilms that are acting like CO2 scrubbers. And they're literally taking CO2 out of the environment and sequestering that as calcium carbonate. And on the flip side, they're dominated by cyanobacteria, which is what Dr. Chris was talking about, and they're giving off oxygen. So I think using microbial communities like stromatolites potentially, and that sequestered calcium carbonate could be used in a lot of different building construction projects, like if we were building habitats or base camps on the moon. So I think that using ecosystems like stromatolites or microbial biofilms could really be very important technological helpers, not only to help with our atmosphere, but maybe giving us building materials. And maybe we want to bring, we just talked about this the other day, invertebrates on the moon too. Maybe you want to start growing oysters and you could use them for food or building materials. So there could be a lot of biological technologies that already exist here that we can harness and use in new ways. Remediating the soil on the moon, that would be incredibly important for growing plants and growing food. So there are a lot of things, lessons learned here on Earth that we can take with us.
Kat has a question. Thanks for this question, Kat. Have there been any significant differences in the types or species of bacteria found on the ISS versus on Earth? Are there bacteria that are somewhat uncommon on Earth that have become common on the ISS or vice versa?
Most of the time, it's the microbiome. So as it really are usually human associated microbes. That's the most dominant one, or plant associated. We're finding more and more plant associated microbes. And as the crew changes over, the microbes are changing out. So when you walk into a brand new space, say you check into your hotel room, within 24 hours, that hotel room has your microbiome fingerprints really on it. So the same is happening on the International Space Station, so or any habitat. So yes, there are definitely microbes that tend to be a little more rare, but most of them seem to be what I call host-associated organisms, because they were transported, by humans or plants that were brought up to the station.
Thank you, Dr. Jamie. If we got to the point of colonizing Mars, for example, and we went through the process of reproducing, that would be for Dr. Egbert, would the children born on Mars have a suitable gut microbiome, or would they have significantly lower levels of gut microflora?
That's a really important question that multi-generational research is just starting. So I'll let Dr. Chris talk to that, but that's a very important issue that people in space biology are thinking about, not just with humans, but with any organism.
Yeah, I know only from the twin experiment with John Kelly, who was 1 year in space, and his brother was on Earth. So they did that too. They checked the microbiome from both of them and how that switched. So it switched in space. I can't really remember what exactly the outcome was, but in microgravity, the gut biome will change and also evolve. There will be an evolution what microbes thrive better with a microgravity issue and all these things. And we know that the microbes have a huge impact on our health too. So yeah, that's a very very large subject of research. What we need to do also about more generations when we colonize Mars. So what happens over generations with the different people, and offspring. Yeah.
So, and it's even compounded for the twin study because they weren't eating the same foods, right? So you don't really know where the microbes were coming from. So it makes it a little trickier. So there's really a lack of evidence. With my squid here on Earth, even when I keep them in captivity, I can only keep them for 1 generation, and then they start to lose fertility. The eggs, so the female mama squid lays a little layer of bacteria to help protect the eggs from getting fouled by other microbes, and that seems to change over time, over generation, if they're in captivity versus. So having the right microbes is going to be critical for multiple generation, and we don't know enough information about that area. So I'm sure with plants too.
Ilan Kusali asked, is it possible to 3D print microbes?
No, not that I know of, but there is some fantastic work being done by a company called TechShot, where they are starting to 3D print entire tissues. They're using cells to, so they're not generating the cell, but they're kind of manipulating the cells and generating these 3D structures for hopefully eventually tissue wound healing and tissue repair in space. But to actually 3D print a single microbe, that isn't, but maybe you meant taking live microbes and building communities. And that's something I think we could do.
Will the altered behavior and division of bacteria affect plant growth in microgravity conditions, since plants rely upon the microbiome in their roots to grow and fix nutrients?
That has to be John.
Yeah, that,
Dr. John.
yeah, the answer is we don't know, but I think that's a really good question, because what you're alluding to is, on Earth, something like, it depends how you count, but like 80 to 90% of plants have symbiotic relationships with bacteria and fungi. And these are really important. In general, the fungi help, for instance, like in phosphorus uptake, which is necessary for biomolecules. And we know very little about the effects. I've done a little bit of work on what we call mycorrhizal fungi, um, which are these involved in helping the plants get phosphate. But we know very little about this topic of the effects of microgravity or altered gravity on these very, very important symbiotic relationships. So great question, but we don't know the answer. We need more research in that area.
Thank you, Dr. John. The last question from Catalin. What do you think of the future capabilities of hydroponics in space?
Yeah. Well, right now that's how they're being grown, is hydroponics. But I think that there's some, what I would like to see happen, this is Jamie with my magic wand here, is that we start working with the soils, lunar soils, and we start looking at how toxic they are, how easily they could be remediated. So maybe we could move beyond hydroponics and actually grow plants in lunar soils and see how that process works. And then maybe you could bring, larger trees, maybe you could bring an orange tree. I'm from Florida, so we just only think about citrus here. And you could start to expand beyond hydroponics, but John probably has much more clear vision of that.
No, I think that's a great answer. And again, when Jamie and I were at this NASA Lunar Symposium, and that's actually a subject looking at, regolith is the term for the soil on other planets. So the lunar regolith or Mars regolith. And there are going to be some robotic landers going up, and some proposed projects are collecting the regolith, and we also have some idea what is in that regolith. Just as Jamie said, she summarized it perfectly, of how could we use that and take out the bad stuff and add some other good things to make the plants grow.
And that's, I was looking at the chat and someone else had a comment about whether we could just forget about growing food and just take nutrient supplements. But I think psychologically growing food and having a garden and having something like that to care for it would be enormously psychologically positive. So I think we should work hard to keep growing plants in space.
I agree. As a plant biologist, I agree 100%. And it's all kidding aside, I've talked to, I've had 8 space experiments, and I've talked to the astronauts, and they just love doing the plant experiments. It's a connection to the Earth, even in low Earth orbit. So you can imagine if you're going to Mars, how important it is. And I used to not think too much about those psychological benefits, and there are some studies, but it's definitely real.
Well, thank you, everyone. I think we covered most questions. Thank you for this interactive, thank you for your participation, interactive questions, and thank you, Dr. Chris, for waking up very, very early, and Dr. Jamie to get on the last minute, and of course for your great moderation, Dr. John. Thank you, everyone. We will welcome you next week for episode 38, Quantum Physics and Quantum Consciousness and the Nature of Our Reality. Is quantum physics responsible for free will in light of consciousness? With Professor Clarice Aiello from the UCLA and Betoni Adams, and another surprise guest. As soon as we get off here, you're able to register for that now. So thank you, everyone, for your participation, and stay well.